So welcome again in another video tutorial. So in this video tutorial we are going to discuss about colony hybridization. So in our previous video we have discussed about genomic DNA library, and in that video I have told you that why you should form a library, right? If you just try to think it normally, we are forming library so that we can get our desired book whenever we are requiring, right? So similarly, genomic DNA library is formed for that similar purpose. Okay, that means it is formed to find out our gene of interest of which we are wanting to study whenever required, right? That means a E. coli genomic DNA library, for example, is formed so that, suppose for example, if we want to study about LAC-Z gene, that is an important gene of black opener because it is encoding the beta galactosidase, right? So if we want to study the LAC-Z gene, then we have to go in a E. coli genomic DNA library, right, to find out the colonies which will contain the LAC-Z gene containing cell, right? Now I want to mention two important things, that is, if we are using any kind of fudge vectors for cloning purposes, then we will get plaques, and if we are using simple plasmid vector or suppose bag vector or any kind of yak vector, there is where there is no matter of infection or lysis of the cell, we will get colonies, right? So depending on that two type of thing, that is the depending on the type of the vector we have used, we can get two different results. In case of our normal plasmid vector or bag vector or yak vector, we will get colonies, but in case of fudge vector we will get plaques, because here the fudge, that is bacteriophage, causing lysis of the cell. Okay, so now let's come back to the topic, that is, so we have to find out our gene of interest containing cell. So now let's try to think it normally, that whenever you go into a library, you will ask your librarian about a book. So this is necessary that you have to know about something what you are requiring. That can be the on which topic the book is written, or who is the writer of that book, or what is the accession number, or anything, or what is the publisher of that book, right? So that your librarian can help you to find out that book. Similarly, just take colony and plaque hybridization as your librarian. Okay, so here the main principle is by which we can find out our gene of interest with the help of our librarian, that is colony and plaque hybridization, is nucleic acid hybridization. Okay, so we have although previously deal with nucleic acid hybridization when we have discussed about southern blotting technique, we have discussed about northern blotting technique, we have discussed about PCR. In such kind of cases we have deal with nucleic acid hybridization, right? So here the same method we will use it. Okay, so as I have told you that your librarian cannot help you if you don't know what you are requiring. Okay, that means you have to know about what is the subject on which you are looking for a book, although, and what is the name of that book, or from which publisher the book is published, or what is the accession number of the book, or anything, right? Similarly, you have to know what is the sequence of your gene of interest. Okay, that is very important. Otherwise, colony and plaque hybridization, which is your librarian in that case, cannot help you. Okay, so first thing that I want to mention, that is, you should know about something related to the sequence of gene of interest. Okay, so on the basis of your the gene of interest, suppose this is your entire genome, okay, and this part is your gene of interest, okay, that is GI. Okay, so and you know what is the sequence of that gene of interest. Okay, so the first step of nucleic acid hybridization is you have to find out a 10 to 60 nucleotide long unique sequence within the gene of interest. Okay, so that is what I have marked here with red color. So this red box is the 10 to 16 nucleotide sequence which is specific for gene of interest, meaning that this particular red colored position is only present in your gene of interest. It is absent in the rest of the part of the genes. Okay, that is similar, like suppose you are looking for any kind of microbiology books, right? So you have to find out the publisher's logo, that of which you are looking the book, that is unique for that book, right? That means suppose you are looking for a Prescott microbiology book, right? So you have to recognize about the McGraw-Hill logo. Okay, that logo will help you to recognize that yes, this is the Prescott, right? Similarly, in case of gene of interest, you have to find out a 10 to 16 nucleotide sequence which is specific for only the gene of interest. Okay, that is the first step for nucleic acid hybridization. Then on the basis of that specific sequence within the gene of interest, we will make a complementary single stranded DNA molecule. Okay, and essentially we will attach a radioactively labeled P32 at its 5' end with the help of T4 polynucleotide kinase. Okay, we have already discussed about this in very first video lectures that we have uploaded regarding recombinant DNA technology. So up to that point, we have developed a complementary single stranded DNA which is of 15 to 20 nucleotide long, and it contains a radioactively labeled P32 at its 5' end. Okay, and it is complementary to the unique part of our gene of interest. Okay, and another thing I want to mention you, that the complementary sequence should not be too much long. Okay, and it also should not be too much small. In both the cases, the uniqueness of the complementary sequence will fall, because remember, the rest of the gene of interest is nothing but ATGC, right? So if you use a complementary single stranded DNA of 3 to 4 nucleotide, then there will be a probability that it will mismatch, right, with rest of the part of the gene of interest, right? So this is, you have to keep in your mind that the complementary single stranded DNA molecule should be 15 to 20 nucleotide long. Okay, so now this complementary probe, I should say this as a probe, will help us to find out our gene of interest containing cells containing colonies, right? Because we have discussed that each of the colonies in genomic DNA library will contain clone of cells that are derived from a single ancestor cells, and each of them will contain multiple copies of a particular stretch of DNA, right? So let's see how colony hybridization will going on. So try to follow what I am saying with the picture. Okay, so this is the mother plate which contain colonies. Colony means here we have used a plasmid vector or bag vector or YAG vector, okay, as a molecular taxi. Okay, so now let's consider that this is a genomic library which contains gene from A to E that I have mentioned here. Okay, and our desired gene of interest is B gene that I have mentioned here. Okay, so now the first thing is that you have to take a nitrocellulose membrane. Okay, this nitrocellulose membrane have some special quality. I want to mention you that this nitrocellulose membrane have special affinity towards the DNA, okay, or also RNA, but here we are mainly concerning with DNA, right? So, and also one thing it is very important here that the size of that nitrocellulose membrane will be equal to the size of the plate. Okay, as a result it will cover all the colonies that is present in the plate. Okay, that is the very important thing that you have to keep in your mind. After that you will use this membrane, that is nitrocellulose membrane, to take a imprint of those colonies that are present in the mother plate. Okay, just like replica plating. Okay, so as a result the colonies will be in our nitrocellulose membrane. Okay, now we will use that imprinted colony containing nitrocellulose membrane to prepare another replica plate. Okay, we will simply imprint those colonies in the another plate containing appropriate nutrient agar medium. Okay, and then we will incubate them to generate colonies. Okay, now after that we will take the nitrocellulose membrane. Okay, it still contains all the imprinted colonies. Okay, then we will treat them with cell lysis buffer. Okay, now what will happen? So let's try to understand. Here all the colonies are immobilized in the nitrocellulose membrane. Okay, and when you apply the cell lysis buffer, they will be lysed at their immobilized condition. Okay, that means they remain immobilized but they will be lysed, and as a result all the cellular materials that is inside the cell will be comes out. Okay, then when you will apply washing step, then all the cytoplasm, all the ribosome, all the cytoskeleton, all the mitochondria that is present inside those cells will be washed away except the DNA. Okay, why? Because at the very first point where during the starting of the topic of colony hybridization we have discussed that nitrocellulose membrane have special affinity towards DNA, right? So instead of those cells, now we have only double stranded DNA molecule at that particular position. Okay, that means the DNA molecule that is present in the cells that are present in that particular colonies will be now there in immobilized condition. Okay, because we have discussed that each colonies will contain same type of DNA, right? So now DNA will be appeared instead of cells, as you can see in this picture. Okay, now we will apply a denaturation buffer. Okay, so denaturation buffer will causes the denaturation of those double stranded DNA in immobilized condition, and it leads to the generation of single stranded DNA molecule. Okay, and then we will wash them all the only one strand will remain. Okay, as you can see. After that, we will add the probe that we have constructed on the basis of the unique sequence of gene of interest. Okay, then we will apply also a renaturation medium. This renaturation medium will help the probe to anneal with those single stranded DNA molecule. Okay, and as we have seen in our southern blotting techniques, here also the probe will bind with those single stranded DNA molecule both specifically and unspecifically. Okay, then after washing technique or after a washing step, only probe who binds with the single stranded DNA molecule specifically remains. Okay, then after that, can you see anything? No. So what you have to do, as you have used a radioactively labeled probe, so you have to use a autoradiography. Okay, so to generate a autoradiogram where we will only have to find out a spot that is nothing but a signal provided by the radioactively labeled P32, that will help us to determine the position of the gene of interest. Okay, because the gene of interest is binded with the probe, right? Now after that, by comparing the result that we have obtained in our nitrocellulose membrane or that we have obtained in our autoradiogram, we will compare this with our mother plate. Okay, and as here our DNA is in immobilized condition, so by comparing the position we can easily find out which colonies contain that particular gene of interest. Okay, and then by lysing those cells we can easily obtain our gene of interest. So that is the concept of colony hybridization. So I will recommend you to watch this video again and again so that it will be helpful to you to understand the entire concept. Okay, so now let's move to the plaque hybridization. So here I do not have to say anything because plaque hybridization is the same that we have discussed in case of colony hybridization. Okay, the difference only this: the plaque hybridization is performed when we have used fudge during the library construction. Okay, which generates plaques. Okay, now I want to discuss with you a interesting concept that the screening of the fudge or the plaque hybridization is much more easier than the colony hybridization. Why? Let's see. I think you know about the basic structure of a bacteriophage, right? And if you know, then I hope you know that our gene of interest inside the bacteriophage is surrounded by only a nucleocapsid, right? On the other hand, in colony hybridization, our gene of interest is within the cell, right? So I hope you can understand that the lysis of the fudge will be far more easier than the lysis of a cell, because during the fudge lysis you have to only apply a fudge lysis buffer, which is nothing but a protein lysis buffer. Okay, but in case of colony hybridization you have to lyse the cell, and which surely contain more heavier constituents than the fudge lysis buffer. Okay, and again after the lysis step there will be a washing step I have discussed. So the washing step will be far more easy in case of fudge hybridization because it contains only nucleocapsid, right? So the crowd of debris will be much more lower in case of plaque hybridization as compared to colony hybridization, because There you have to deal with ribosome, mitochondria, you have to deal with cytoplasm, you have to deal with inclusion bodies, you have to deal with cytoskeleton. In contrast, in plaque hybridization you have to only deal with those nucleocapsid. Okay, that means washing step will be very easy for fast plaque hybridization. Okay, so that was the concept regarding phage hybridization and colony hybridization. And another thing I want to mention you, that during the plaque hybridization, when you will create a replica plate by imprinting those colonies that are already imprinted in the nitrocellulose membrane, you have to imprint them within a bacterial lawn. Okay, just try to think about this, because here we are willing to grow phages, right? And what is the food of the bacteriophage? Nothing but the bacteria itself. So we have to provide bacteria as a food material. That we have done during the replica plating formation in case of colony hybridization, because here we are growing bacteria, right? So there we have provided a appropriate agar medium, right? Similarly, in case of growing of phage, we have to provide bacterial lawn so that they can grow and develop plaques. Okay, so that was the idea regarding colony hybridization and plaque hybridization. So I hope this video will be helpful to you, and thank you for listening to this class.